Assalamu alaikum. Hello everyone. Welcome to the third International Conference on Applied Zoology, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. I'm Syed Rahman Shah, belongs to Department of Zoology, Kohat University of Science and Technology, CAST, Kohat. I completed my work under the supervision of Dr. Farmanullah on the topic effects of atrazine on blood biochemical and hematological indices of Grass Carp. Introduction. We are familiar that Pakistan is an agricultural country where different herbicides and pesticides are used for the control of herbs and pests. So here, atrazine is also one of the commonly used herbicides used for the control of annual grasses and broad-leaved weeds. Atrazine is not degraded by microbial or hydrolytic process. So because of that, atrazine is present in the soil due to irrigation and water or raining, the atrazine runoff into water bodies. When it runoff into water bodies, so we know they are aquatic animals, and we also familiar that atrazine is highly toxic and carcinogenic in nature. So it have adversely effect upon the immune response, blood indices, growth, reproduction, behavior, and physiology of the fish. What was the objectives of the current study? So there was two objectives in mind. The first one was to analyze the potential effects of atrazine on biochemical indices of grass carp, and the second was to investigate the potential effects of atrazine on hematological indices of grass carp. Material and methodology. I completed my work in the two steps. And the first step, collection and acclimatization of grass carp. Grass carp was collected from the local fish farm from Peshawar and then transported to the Aquaculture and Fishery Laboratory, Department of Zoology, Kohat. After that, the fishes were acclimatized in the water tanks for 2 weeks. And after that, atrazine dilution, technical grade, atrazine 97.5% were used for the stock solution, in which 5 ml of atrazine and 5 ml of 80% acetone was mixed. And after that, from this stock, 1 ml of the solution was taken and it was mixed with 9 ml of distilled water. From that 10 ml solution, different concentration was given to the fish. So we made acute toxicity experiment, and that experiment we made 5 groups. One was the control, and the other 4 was the atrazine treated groups, in which different concentration, 50, 100, 150, 200 microliter per liter were given to the fish. After that, and the second step, we find first the LC50 or lethal concentration of 50 on the 50% population of the fishes were determined. And also the hematological and biochemical component was also determined. So going towards the results. And the results, I will only share with you 2 components from the hematological and 2 components from the biochemical due to shortage of time. So the first one is RBC, red blood cells. And the graph, you can see that the red blood cell concentration in the control are different from that of the treated groups and it significantly decreased. The reason behind it is because atrazine is toxic to the fish and it affects different cells of the fish, behavior, physiology, biochemistry of the cells. So because of that, the hematopoietic tissues also got affected because of the toxicity of atrazine. When they affected, so it not produce more RBCs into the bloodstream. So because of that, the RBCs concentration, the RBCs level decreases, and different treatment groups with different concentration and different times. The second one is the white blood cell WBCs, and here we see in the graph WBC level was increases from control to the different groups significantly increased, because when the foreign particle or the foreign toxin enters the body, so the lymphatic system produces lymphoid cells, and these cells fight against these toxins. So here WBC level increases, which means that this WBCs fight against the toxin and its level was increases. The third step is the glucose the third component is the glucose. And we see in the graph that the glucose level was significantly increasing. Why it is increases? Because we are familiar that blood glucose considered as indicator for stress response in fish. Hyperglycemic condition induced by environmental pollution might produce stress response in fish by the inhibition of the gluconeogenesis process, which enhances glycogen breakdown in the liver and consequently elevates blood glucose level. Another reason may be the hyperglycemic state could be explained as result of the increased secretion of catecholamines from the adrenal medulla, which in turn enhances the breakdown of glycogen, and because of that, the blood glucose level will increase. Why it increased? Because of the toxin atrazine given. So it's all about the glucose, and the graph we can see a significant increase was come. The last one is the cholesterol. And from the graph, we see that the cholesterol and the control group was increased and it was then significantly decreases. What's the reason? Because we are familiar that cholesterol is the best material for all steroid hormones. When it's increased due to its cortisol synthesis, then a large amount of cholesterol is needed. Therefore, the reduction in the amount of cholesterol may be related to its utilization and the manufacture of cortisol arising from stress created by the toxin atrazine. And because of that, we see that the cholesterol level was decreases because it was used in the cortisol synthesis. Thank you so much for your attention.